Good morning. Welcome back to Biology 1302. We're going to talk about Chapter 8 this morning, DNA, Inheritance, and Biotechnology. Specifically, this chapter talks about DNA replication, both through a process called binary fission and a process called mitosis. No living organism can reproduce without cell division. If you're a unicelled organism, you have to reproduce. If you're a multi-celled organism, you likewise have to reproduce at some point. So to do that, the cells have to replicate themselves, and we're going to talk about how that happens. Cell division produces a continuous supply of replacement cells in multicellular organisms. In the process for unicellular organisms, it's the way they reproduce. These particular cells right here, again, when you see the, whoops, back up, sorry about that. When you see the word false color, that just simply means that the slide has been either dyed to make it a certain color, or it's a computer-generated color. This would not be the color that the tissue or the cell is. This shows the reproduction of chromosomal material, which we're going to talk about in a little bit. One of these would be the original set. The other is the new set that's going to go into the new cell. Sexual life cycle, eukaryotic cells divide through a couple of different processes. We're going to start right here. Fertilized egg, which is here. You see this is the egg cell. There's the sperm. There's the fertilized egg, which is referred to as a zygote. That cell will begin to undergo what is called mitotic division, or the process of mitosis. That's the way cells are going to reproduce, produce first multiple examples of this cell. Later in the semester, we will talk about a little bit about human development, and we'll talk about what happens when the cell goes from one cell to two, from 2 to 4, and then 4 to 8, and you keep that replication going until you get about 500 cells, and then they start to develop a little bit differently. The fetus grows and develops into a mature adult, consisting of countless numbers of cells. We have male and female here. Then there's another process which we will talk about in the next chapter, I believe. It may be later on in this chapter. This is the first time I've taught general biology in a few semesters, so the textbook may have altered a little bit. But either this chapter or the next one, we will talk about meiosis. But ultimately in males, the process of meiosis is going to produce the sperm. In females, it's going to produce the eggs. These are collectively referred to, again, this is one of those words that has a couple of different pronunciations. One of them is gamete. I was taught gamete when I went to school. When I was taking my education, my professors said gamete. At other places, and again, I think maybe this is a symptom of where you're taking your education. If you're in the South or maybe in the West, they say gametes. On the East Coast and maybe in Europe, they say gametes. But whichever one of those terms you are saying, we're referring to reproductive cells. These are the cells that are actually reproducing a multi-celled organism. Each mature individual produces sex cells by another form called meiosis, which again we'll talk about in a later chapter. Meiosis only occurs during reproduction. So we will talk about that as we get to it. Sexual life cycle, gametes fuse, again in the process called meiosis, to contain half the amount of DNA. So this man's cells have 46 chromatids, 23 pairs of chromosomes. This lady likewise has 46 chromosomes, 23 pairs. Ultimately, when a sperm is formed, a sperm only has 23. Basically, the way this works is you have chromosome 1 and chromosome 1. You have 2 copies of chromosome 1. You have 2 copies of chromosome 2, et cetera, et cetera, et cetera. Then when meiosis occurs, those are separated into 2 different cells. You have 2 sperm cells, one that will contain 1 chromosome 1, and another cell that will contain the other one. So at fertilization, goodness gracious alive. At fertilization, the zygote inherits DNA from both gametes, and there you get one set of chromosomes from your mom or your dad and one set from your mom. Mitosis, on the other hand, has lots of different roles. Here we start. This is an embryo. Oh, darn it. This is an embryo. Still learning to use the laser feature on my pointer. But this is an embryo, very early in development. You can see if you look at this, and we'll talk about this in the human development area. We have this baby as it's growing. You see a very large head in relation to the body. As the baby develops, the body will get larger. The head will not reduce in size, but that difference between or that ratio of head size to body size changes as development occurs. Here we have a young boy playing soccer, or as they say as they say in the rest of the world, football, and he scraped his knee. The process of fixing and repairing that damage will be a mitotic cell division. Here we have a salamander who's lost his tail. That tail will eventually grow back on some of these animals. You can see it's already starting to grow back a little bit. That cellular reproduction to make a new tail is a mitotic division. And again, not real sure what that's supposed to be, but you see you've got an individual cell here and another cell here, and those are reproducing by mitosis. That's probably, since it says some organisms reproduce asexually by mitosis, I would suspect that is some sort of a one-celled organism. Cell death is part of life. The process of cells dying is referred to as apoptosis. It's a process that can carve out distinctive structures depending on development. Here we have a duck and a baby chicken, and you can see early in development their feet look fairly similar. If you actually go back a little bit further in development, they would actually look more alike than they do here. But you see what happens to the duck's foot, forms the 1, 2, 3, 4 feet, but you see they're webbed, which enables it to swim. But over here on the chicken, the feet are separate, the toes are separated. And the reason that is because evolutionarily, chickens fed by scratching the ground. And if they were trying to scratch the ground and uncover the things they need to eat, that webbing would really get in the way. So again, you've got these cells that produce this die over here through this process of apoptosis, so that webbing never forms. There are cases where baby chickens, due to a mutation or some sort of mess up in genetics, will be born with webbed feet because apoptosis did not occur correctly. There have also been cases of ducks being born with a foot that looks more like this because apoptosis occurred when it really wasn't supposed to. Let's skip over the mastering concept. So DNA replication. Before cells can duplicate, they must replicate. If you're going to make a new cell, you're going to have to make all the components inside that cell that's needed for 2 individual cells to live. Now, there's enough cytoplasm, there's enough mitochondria, there's enough Golgi, there's enough, all of those things there's enough of. The thing there's not enough of is DNA. There's not enough DNA to support 2 cells. So before a cell can think about dividing, dividing, it must copy or replicate its DNA. The DNA must duplicate. So the entire genome is replicated so that each new cell gets a new copy of the DNA. And again, here's that picture of the DNA down here, another set of DNA over here. And again, this is a scanning electron microscope. This scale right here, it says it's approximately 5 micrometers. A micrometer or a micrometer is 1/1,000,000th of a meter. So if you took a standard meter stick, and if you look at a standard meter stick, these little individual marks, like right here it says 80 and then 81, 82, 83, 84, Those are called centimeters because there's 100 of them in a meter. Each little line between 80 and 81 on a meter stick represents a millimeter. Now, if you take the distance between two of those lines, those little bitty distance here, you know, it's not a whole lot thinner than my fingernail is thin, not my fingernail, not this way, this way. If you take that little distance and slice it into 1,000 pieces, that's a micrometer. So very small, so you can see how much this must be magnified if this distance is equal to 5 1/millionths of a meter. Excuse me. So DNA is made of two chains. We've talked about DNA before. They're complementary to each other, A to T, C to G. Each strand acts as a template. They're both used to build a molecule of DNA. So what will happen is this side will separate from this side. This side will be used as a template or a pattern to produce a new DNA. And this side will be used as a pattern. And if you will remember, in a previous chapter, we talked about that the DNA had one strand going one way and one strand going the other way. I really like what this author and this illustrator have done. And this is actually the first time I've ever really noticed this. But if you will look on one side, the letters are correct. They're up there. They're in the right configuration. If you look over here, the letters are upside down, showing you that this strand is going this way, but this strand is going this way. So one strand going one direction, one strand going the other direction. DNA replication is what is referred to as semi-conservative. What semi-conservative means is that when replication is done and you've now got two new pieces of DNA, each new piece of DNA is going to contain one copy of old material, or the original material, and one copy or one side that's new material. So if you call, let's back this up. If you call this side A and this side B, and then the new sides X and Y, then you would have A X and B Y, because you're getting half of the original material is being maintained. And that is semi-conservative. So we have one parental strand and one daughter strand. And again, this shows, I'm not sure how well you can see it on the screen, but these two strands on the inside that are labeled daughter cells are a lighter blue color than the outer strands, which are the parental strand. Enzymes play a role in this. There are certain enzymes called helicases. The job of the helicase is to unwind the DNA. So you see here it is in its coil pattern. There's the helicase. The helicase is unwinding it and also serves to unzip it. It looks kind of like a zipper the way they've got it drawn there. The complementary bases are then added to the growing strand of new DNA. And again, you can see on this one, you've got the light blue here, over here, you've got the light blue this way, and then it will eventually form some more here. And this one is going to grow continually. This one's going to grow in small little pieces. The enzymes responsible for making new DNA, putting the new bases on there, are the DNA polymerases. And then this side, I'm going to check right quick and see if we can see the little, nope, it doesn't show them very well. But what happens on this strand is this little piece will be made, and then another piece will be made, and another piece will be made. But as each piece is made, this piece will not be connected to this piece. There will be a gap right there where the bonds haven't formed. And it is the job of the ligases to join those short strands of DNA. Those short strands of DNA are called Okazaki fragments. It was a Professor Okazaki that figured out this discontinuous, called discontinuous replication. This is continuous. This is discontinuous. let's step back a minute, and I thought it would go with a little bit more about semi-conservative. And there may be a slide, I just simply don't recall, but there may be a slide later that talks about semi-conservative. But why is semi-conservative important? Semi-conservative is important because if you always maintain the parental strands, the chances of errors are greatly reduced. The possibility of making a mistake is greatly reduced by always maintaining that parental strand and the daughter strands separately. DNA replication requires the use of energy. Again, this is where we're getting our old friend ATP, adenosine triphosphate. As this phosphate, third phosphate, is removed, that's what helps the attachment of the DNA. So ATP is required for the reactions of replication, and energy from ATP is used to produce nucleotides, unwind the DNA, and build new strands. So all of the reactions happening in building up DNA or building up the new DNA molecule is under the control of ATP. Replication enzymes, again, you're going to see that we have parental strands here, shows the origins of replication. The strand will get opened up, daughter strands will form, and again, here you see these little discontinuous areas that are eventually linked together. DNA replications begin simultaneously at different spots along the chromosome. Think about it, the chromosome may be as long, as this board is, for example. So it's going to start here, it's going to start here, it's going to start here, and you're going to have one strand going this way, and one strand going that way, and one strand going this way, and one strand going that way, et cetera, et cetera. And they will eventually come together to form the two parent strands. Important to understand that it doesn't just simply start down here at the end and then work its way that way. Because if it started down here, the strand that has to run that way would have nowhere to go. That's why it has to start here. Let's see. In bacteria, prokaryotes replicate in a very similar way, but since they are prokaryotes, we refer to it by a different name. We refer to it by a different name, and we call it binary fission. Fission sort of means to split. Binary means two. So here we've got a single cell with its cell membrane, cell wall, and its DNA. Now, again, you can see the DNA start to replicate. Now you've got two copies of the DNA, and the cells separate through a process called binary fission. Again, one of the things we have discovered over the last, probably 50 years or so, is that bacteria especially, but prokaryotic cells do have a process for transferring their DNA between each other. For many years, it was thought that bacteria were simply cloners. They cloned themselves. Each bacterial cell made a clone of itself when it separated or when it divided. And the only way new information would come into that was through mutation. Then it was discovered that bacteria actually do have a way to sort of share DNA. What they will do in bacteria, and again, the archaeas do this as well, some donor cells can directly transfer its genes to a recipient. So here you've got the original cell. It's made its DNA. It's made a copy of its DNA. But instead of this cell separating, what it does is it attaches itself to another cell of the same type by what is called a sex pilus or sex pili. The new DNA comes over here, and so now we have the recipient cell producing the same excess DNA as the source cell. Again, shows that this can happen. DNA can produce DNA fragments, and then the recipient cell can take up some of that DNA, and you'll see the little lighter color right there. Two different ways that this can happen. You can transfer a whole DNA, or you can only transfer parts of the DNA. Eukaryotes wind up, they're extra tightly. You see, here's the loosely packed chromosome, looks like this. Here's the condensed DNA in this. In lab, if you're taking an on-campus lab or you're doing a virtual lab, you will probably at some point be asked to look at slides and find cells that are in this form and cells that are in various stages of that form. DNA is unwound in replication, but afterward it condenses before it starts to divide, or it coils up. So after replication, DNA will look like this. As the DNA gets ready to duplicate and divide, it will condense and start to look more like that. Chromosomes and eukaryotic cells are almost always represented by an X-shaped structure. This is one chromosome. This is another. This is, say, chromosome 5 you got from your mom. This is chromosome 5, the reproduced copy of it. The structure that chromosomes are made of is a product called chromatin. When you tightly pack the chromatin together into these structures called chromosomes or chromatids, then they are finally referred to as chromosomes. Each chromosome will again consist of 2 identical sister chromatids, 1 for each copy of the replicated DNA. So this is my original 5 I got from Dad. And this is the new 5 I got from Dad forming, and you see it starting to condense and bring itself together. Before replication, DNA is visible only as chromatin. You can see that structure inside that nucleus is very loose. There's no real structures visible. The DNA will be unwound or straightened out. It will start to replicate. A centromere will form, holding the 2 strands together. The new strand will be forming from the old, and finally you wind up with the full-fledged replicated chromosome. And the A, B, C, D, E, and F structure here are representative of genes that are along that chromosome. Maybe they control hair color, nose shape, ear shape, eye color, etc. And here you can see again, that's the photomicrograph of nucleus that is unwound, and here is one where you see the chromosomes are now tightly bunched together. Mitosis, again, generates exact copies of the original. Here's my original cell. It's going to go through prophase. In prophase, what has happened is the DNA has duplicated. Duplication is complete. The cell membrane that's holding the nucleus together, the nuclear membrane, has started to break down. These structures called centrioles have started to form. These structures called spindle fibers will begin to form. During metaphase, the nuclear membrane has completely broken down. The chromosomes have now aligned themselves in those sister chromatid pairs along the center of the cell in a process called metaphase. The cell moves into anaphase, where the centromeres break down. Centriole, centromere. Centromere breaks down. What's holding the 2 sisters together is the centromere. It breaks down, and the 2 sister chromatids are allowed to move independently of one another. They will begin to move toward opposite sides of the cell. During that process, the cell will begin to elongate. During telophase, this is another one of those words that you may hear people pronounce telophase. I learned telophase in school, but I also answer the telephone. I don't answer the telophone. So I say telophase. But again, the cell has elongated a little bit. New nuclear membranes are starting to form. The DNA at this point is actually starting to unwind, but the authors don't usually draw it that way. And then you have a split, and you have 2 independent cells forming. And if you look very closely, this cell looks exactly like that cell if you take away the duplicated chromosomes. See, you only see 1 chromosome in each pair. Here you see 4 pairs. Here you only see 4 individuals. You see 4 individuals here, but you see 4 pairs. That's because one of each one of them has gone to this cell. One of each one of them has gone to this cell. Again, this cell is showing duplicated chromosomes. This cell is showing chromosomes not duplicated. The cell cycle is a repeated process. It's a series of events. This is the life cycle of a cell. G1, GS, and G2 are referred to as interphase. Interphase is where the cell is going to spend most of its time. Most of the time, the cell's doing the things it's supposed to do. Sweat gland cell making sweat, kidney cell filtering urine, whatever. Most of the time, the cell is going to be in interphase. Then when the cell gets ready to reproduce, DNA replication is going to occur here. Any last additional growth and preparation for division happens here. And then the cell will enter the cell division stage, more commonly referred to as mitosis. Textbook authors and illustrators always show these phases: normal cell growth and development, DNA replication, and additional growth. They always show them sort of equal sized in this little pie chart thing, and that's actually not accurate. This one should actually be significantly bigger than either one of these, and then this one should be bigger than this one. This G2 phase doesn't last very long at all. This is by far the longest phase, but no matter what artist rendering you look at of the cell cycle, these three stages are always going to be depicted as being equal. In mitosis, we enter the prophase phase, which we just talked about, then metaphase, anaphase, telophase, and then one cell is going to start this program again. Another one, cytokinesis, is going to occur. That's where your two cells are going to be formed out here, and then both of those new cells will start this process again. During interphase, we talked about carrying out their normal everyday functions unrelated to cell division. During G1, that's where the cell grows and functions properly. It's getting ready to divide. It will move into the S phase. During the S phase, the DNA will be duplicated. The entire genome will be copied. Every chromosome will be copied. It is then ready to divide. And during G2, any specialized proteins that are necessary for production, if the cell needs to create a couple of new Golgi or a couple of new mitochondria or whatever, that's when these are going to be made. The mitosis phase, again, we talked about the stages of mitosis. That is the cytokinesis. Another thing that is often done in textbooks is cytokinesis is listed as a stage of mitosis, and it's really not. It is a process unto itself. It's talked about here, and it's lumped in with mitosis because ultimately at the end of mitosis, cytokinesis occurs, but it's not technically part of mitosis. Again, here's the mitosis steps. We showed them earlier. You've got prophase, metaphase, anaphase, telophase, followed by cytokinesis. I would suspect on the exam covering this material, there will be a nice little question that says, give me the order of mitosis, or give the stages of mitosis in order. That will be, the answer will be prophase, metaphase, anaphase, and telophase. If you include interphase and cytokinesis, you will not completely miss the question, but you will not receive full credit because, again, interphase and cytokinesis are not technically part of mitosis. Interphase is the events preceding mitosis. Cytokinesis is what happens after mitosis. Again, if it's say a 10-point question and you say prophase, metaphase, anaphase, telophase, you get 10 points. If you say interphase, prophase, metaphase, anaphase, telophase, cytokinesis, you will probably only get 7 or 8 of the points. I won't, give you 0, but again, extra information. Answer the questions that's there. Don't give me extra information. Again, during prophase, we talked about this. The chromosome, the replicated chromosomes condense. This is again showing an early prophase, late prophase example here. You've still got the nuclear membrane. The spindle fibers haven't formed. Here, the spindle fibers have formed on the centrioles, and the pairs are free to wander, but they haven't really attached themselves yet. Then in metaphase, everything is attached in the center. Again, you see how they form a nice little line up and down the center. That looks like an onion cell, maybe, that they've got drawn here. That's usually what they show. Then we have anaphase. Again, we have anaphase. You see they've separated. Half of them have started to move one way, half of them have started to move the other way. Telophase, two new cells are now fixing to form. We've got new nuclear membranes forming. We've got the individual chromosomes. They're going to start to unwind. This contractile ring will form. A cleavage furrow will form. And eventually, this is the cytokinesis process, the formation of the contractile ring and the cleavage furrow. Here you can see that, again, yes, I'm pretty sure now that is an onion cell. And then early interphase will start once these two cells completely separate. There's the cytokinesis step. And again, actual photomicrograph of an animal cell of some sort. I have no idea what that is. But again, you see the cleavage furrow, the contractile ring, and the nucleus beginning to form. Cells in plants have to do things a little bit differently. If we look back here on the animal cell, when you, hang on, let me get back to where I need to be. Okay, on the animal cell, you see this just has to pinch in, the contractile ring forms, the contractile ring pulls this side toward that side, it contracts, ergo the contractile ring, the cleavage furrow will form, and then eventually these will come together and pinch apart. In a plant cell, however, plant cells also have a cell wall. So a cell wall has to form. And that is done through vesicles. In the cell wall, or in the cell, the components that make up cell walls will be packed into vesicles. Those vesicles will move to the center of the cell, and they will sort of line up along the same line where the cells, or excuse me, the chromosomes were lined up during metaphase. During metaphase, the chromosomes were lined up at a particular place along these these microtubules here, the spindle fibers, and the vesicles containing the components of cell walls will line up here. When those vesicles start to attach together, the components found within them will start to assemble themselves and eventually will form a new cell wall, where here you have one cell with that, just a demonstration of a wall, a structure called the cell plate will actually form, and then the new cell wall forms as cytokinesis is completed. The cell wall, or excuse me, the cell cycle is under very tight control. There are checkpoints all along here to make sure things go right. Checkpoints ensure that all the DNA has been replicated. Until all the DNA has been replicated, the cell will not move out of the S phase. Make sure that DNA is not damaged. During this point, during G2, the DNA will be scanned or checked to make sure everything is where it's supposed to be. And DNA polymerases, in addition to putting everything together, also can correct mistakes. If there's a mistake made, DNA polymerase can fix it. Where did we go? And finally, they make sure that chromosomes line up and separate correctly. Cancer cells are cells that are dividing out of control. In cancer, the body loses control of cell division. These steps here aren't working correctly in cell division of cancer cells. When cells start to divide erratically, erratically is a good word, they can go two different directions. They can form what is called a benign tumor, or they can form what is called a malignant tumor. Benign tumors have a capsule on them of connective tissue that keeps the tumor from spreading. If you watch Dr. Pimple Popper I think she's on Discovery Channel. This is a lady out in California. She's a skin doctor, and people go to her when they have these really strange, weird growths. You know, a guy's got an orange-sized thing growing out of the side of his neck that for some reason he has ignored for 10 or 12 or 15 years, and now he said, once it gets to be the size of an orange, he thinks it's time to get work done on it. My wife and I sit there watching this going, why did he wait so long? Seriously, why did you wait so long? But when she opens those up and can squeeze and all of that mess and gunk and gnarliness comes out, those are almost always benign tumors because they are just compact. There's a capsule on them. And as the cells in that tumor continue to grow and become more and more and more and more and more, that capsule continues to form and it keeps all of those cells encased in that one place. That's a benign tumor. What happens in malignant tumors is that they can seed new tumors throughout the body. Here, this one's growing, but if you look very carefully at this drawing, you see there's a capsule around it. There's no capsule here. And what happens in a malignant tumor is cells can break off of it and either enter a blood vessel or a lymph vessel, and then they wander around where they then go out and form new tumors in other parts of the body. If you've ever heard of anybody, say, having lung cancer, but they have lung cancer, but it's in their brain. The bike racer who I used to defend, I used to defend Lance Armstrong. In my class, I would defend him, saying, that the chemotherapy he did altered his body chemistry, and that was the reason he was able to do the things he did. Yeah, we all know now he was a great big cheater. But he, at one point, was given 6 months to live because he had testicular cancer. But where was his testicular cancer found that was going to kill him in 6 months? It was in his brain. He had testicular cancer, but it had metastasized to his brain. The movement, the movement of these cells from the origin to new areas is called metastasization. It's said to have metastasized. These cells, either these or these, are dividing when they don't need to, and they override those checkpoints that we talked about earlier. In both of these cases, cells are dividing primarily more often than they should. These cells around this will say they're dividing every 7 to 10 days. These are dividing 3 or 4 times a day. So their rate is much increased. A tumor isn't simply an abnormal mass of tissue. They can either be benign or malignant. We talked about that. Again, here's a little blow up, a little bit bigger version of what we just saw. Cancer treatments include surgical removal of the tumor. With today's technology, if a tumor is operable, in lots of cases, the outlook is pretty good. However, if the tumor is not operable, if it's invaded tissues to the point where we can't get it all, or it's invaded vital tissues to where it can't be operated on at all, prognosis many times not so good. There are anti-cancer drugs that can slow or block division of cells. The problem with that is most of them, as they're slowing and blocking the growth and development of cancer cells, they're blocking the growth and development of all of our cells. So we're looking at killing, you're slowing down good cells and bad cells. Radiation, again, it says that kills targeted tumor cells. The way radiation works is we try to pinpoint our radiation, well, it would be here, right here on this tumor. The only problem with radiation is it's not as precise. It's better today than it was 30 years ago, but still, when we radiate this area, we're going to damage some nearby good tissue as well. You can reduce your risk of cancer by doing several things. Number one, eat healthy, less saturated fats, more fruits and vegetables. We know that there is a relationship between saturated fat and certain types of cancers. We know that. Again, some people will argue, well, that's a correlational study, and correlational studies don't give you cause and effect, and I know that's true. But, when you look at certain colon cancers, and 95% of colon cancer people have their entire lives consumed large amounts of saturated fat, a much higher percentage than the general population, then that is a positive relationship. Obesity, maintain a healthy body weight. Get regular, vigorous exercise. Again, exercising stimulates all sorts of things. It's not just to get buff. Regular exercise releases endorphins. Endorphins make you feel better. Endorphins play a role in the immune system, making the immune system work better. So lots of benefits to regular, vigorous exercise. Tobacco use. I'm here to tell you there is no safe way to take things into your lungs that are not meant to be there. So if you're a smoker, if you smoke a pipe, if you smoke a pipe, if you smoke a cigar, if you smoke a cigarette, none of those, there is no healthy one. Vaping. Your lungs are not designed for you to inhale mists of any kind. Now, yeah, there are mists that medication can be administered that way, but the chemicals you are getting from a vape pipe are different than cigarette smoke, but they're absolutely not healthy, and they're not they are not unharmful. Tobacco use. There is no safe tobacco use. I don't smoke, I dip, Mr. Russell. Okay, then you want to have oral cancer and you want to have your teeth to fall out. And there are some wonderful pictures of people who have gigantic holes in their mouth right here that they have to wear prosthetic jaws because their own jaw has been eaten away by the cancer. Tobacco, but again, and I have this conversation, especially with some of my younger students throughout, the last 20 years of doing this. Well, if tobacco's so bad for us, why won't the government ban it? Why won't the government ban it? They make too doggone much money off of it. And we could have a whole, we could spend 2 hours talking about the social ramifications of tobacco use. Where are all the tobacco stores? Where are all the tobacco outlets? They're in the poor parts of town. You're not going to go up into, the rich high dollar parts of town. I pick on Hillcrest and Little Rock, and I don't mean to pick on Hillcrest. That's not what I'm saying. But yeah, go up and drive around Hillcrest. There's not a tobacco store up there. Go down South University and see how many tobacco stores you see. Tobacco stores are always in the poor neighborhoods. So no safe tobacco. Ultraviolet radiation, the light coming out of these lights above my head. Why do they have those funny shaped little grids over them with the little sort of diamond texture? Those are called filters. They're supposed to be filtering the UV light so the safest of the UV is coming through and the other parts of the UV. Now remember, the UV spectrum's this long. This end down here is safer than this end. So we want to use this end down here, not this end over here. Cancer-causing viruses, the human papilloma virus associated with cervical cancer. Use condoms to avoid sexually transmitted diseases. Again, there are a multitude of viruses. Self-tests and medical exams for early detection. One of the reasons, you're an African American male and you're in your 40s and 50s, good idea to be checked every so often for colorectal cancer. All men should do it, but especially African American men, because there's, for whatever reason, there seems to be a higher concentration of colorectal cancer in African American males than there is in other ethnic groups. So detection time, found early today in 2021, found early. Most cancers, with a few exceptions, are I'm not going to say easily treated, but they can be treated with some effectiveness. And then your family history can't be avoided. But what you can do about family history is, ladies, if your mother had breast cancer, your grandmother had breast cancer, your other grandmother had breast cancer, and all of your aunts had breast cancer, probably a good idea to you to stay on top of your self-exams and see your breast doctor occasionally for a mammogram. Again, you can't do anything about it. My family's always been heavy. Nothing I can do about the genetics that led me to be heavy, but I know you can't really tell much about. I've got a big floppy shirt on. I've lost 125 pounds. At one time I weighed almost 300. I've actually lost more than that now. because I weighed almost 340 pounds. I'm now down to 202. Thank you very much for your applause. And part of the reason that I did that was because I have a family history of certain types of cancer. I also have a family history of heart disease. I don't want to die at 57 like my uncle did. Beat that, because I'm 58 now, so I can't die at 57. But I also would like to live a little bit longer than my dad did. My dad lived to be 76. I would like to live longer than that because in April of 2020, I adopted a 1-year-old baby. She's now 2. She's about yay high. You can't see my hand because she comes to about my knees. But her name is Lindy. I will talk about Lindy a lot for the rest of the semester. And I plan to be around for Lindy. So I lost weight. I got my diabetes under control. I'm exercising regularly. I'm not spending all of my weekends sitting in front of the television anymore. I'm going out and working in the ground, working in my yard. I'm growing a garden. I'm eating more fruits and vegetables. The last 2 or 3 weeks, I have not been eating as healthy because due to some outside forces, I have been having to consume more fast food than I really like. But there have been things going on to where the wife and I just simply didn't have time to sit and cook a decent meal. We just had to run up to the McDonald's and get some chicken nuggets and a burger, and we had to make do. But hopefully things have calmed down now. I've got all of my work issues out of the way. I've got my schedule for next semester done. I've got my budgets done. I've got all of those chair responsibilities that I have to do done. So hopefully things can settle down around my house, and we can get back to the healthy diet. Investigating life, the race against cancer. Again, every tumor contains a mix of drug-resistant and susceptible cells. Chemotherapy kills the susceptible cells and changes the balance power. Free from competition, the drug-resistant cells reproduce rapidly. So adaptive therapy is a new approach. Basically, at this point, the blue line represents standard therapy. The purple line, skipped doses of therapy. You have the resistance cells being the red ones, the susceptible cells being the green ones, and then you have declining doses over time, and you can see the tumor volume. If you run the standard therapy, what you see is here, because you're getting more and more and more of these. If you skip doses, you see what happens very quickly, and now you're up here, way up here, even higher. But if you have a declining dose, basically what you're doing is you're knocking out a bunch of these, but then you decline your dose, and so you don't greatly increase the resistance of this. And hopefully, if this can be continued in this the sort of upping and down and up and down and up and down, you can eventually get rid of those cells and get you to a point of what is called what is the word? What is the word, guys? When your cancer's not there anymore. Remission. That's the word, remission. All right. If you have any questions, text me, or not, don't text me. Email me in either the left-hand side of the Blackboard shell, open contact, send me a message in Course Messenger, or just send it to my trussell@uaptc.edu. It's Friday where I am, so have a great weekend. If you're watching this on Sunday night, get a good night's sleep and have a great next week, and I will see you later.